Hello, my name is Dick McIntosh. I'm a cell biologist from the University of Colorado. I study cell division and have been working on that problem for about 40 years. It's a fascinating problem because cells must divide in order to live. Cells are very complicated, so the division process is itself complex because every time a cell divides, you basically have to build two new cells where you previously just had one. This means, of course, there's a lot of biosynthesis that must go on in order to provide the materials that will allow a cell to produce two viable daughters. Now, cells are very complex. Their complexity can be thought of in terms of the number of instructions that it takes in order to build a cell. And although we don't know that number precisely, you can estimate it from the number of genes and from the ways in which genes are regulated and from existing proteins in cell. And in short, the number of instructions that's involved to build a cell is more than the number of instructions that it takes to build even a very complex man-made object, such as a moon rocket or a supercomputer. This means that cells really are at the very high end of complexity in terms of the kinds of structures and systems that we think about. But on the other hand, cells are very small. They're so small that if you take cells from, say, the human liver, it takes something like 1 million of them to make an object as big as a pinhead. And this means they're not only highly specialized in terms of their complexity, but also in terms of their micro-miniaturization. Now, on top of this, cells can reproduce themselves. Imagine what you would have if you had a self-reproducing moon rocket or supercomputer. But cells can achieve this remarkable goal, and this is the process that we're going to look at in 3 lectures. In this first one, I'm going to give background information. To some extent, it's textbook information, but I hope I can put an interesting light on it for those of you who already know quite a bit about the subject. In the second lecture, I'm going to describe experiments that have been done by a wide variety of laboratories, including my own, in order to try to probe the machinery that allows the cell to divide. And in the final lecture, I'll talk about some recent work from our lab in which we're trying to understand the actual mechanism by which chromosomes move to the poles when a cell is dividing. So, let's start thinking about this in terms of the structures that you have in a cell that have to be reproduced. If you look at this electron micrograph, you see, of course, the familiar nucleus, the cytoplasm with all its complex membranes. Not shown here are all the complexities of the cytoskeleton as well. All of these structures that we can see in a cell are actually made from the assembly of macromolecules, proteins in most cases, sometimes nucleic acids, carbohydrates, and lipids. And all of these constituents have to be synthesized before the cell is ready to go into division. So, preparation for division involves a tremendous amount of biosynthesis. The instructions for this division come by and large from the DNA, which is located in the nucleus and has the familiar base pair structure that allows the sequence of the nucleic acids to define the sequence of a message, which in turn defines the translated product. And yet, that complexity of itself is not sufficient for building a cell, because you could take DNA and put it in a test tube and transcribe it and translate it, and you still won't build a cell. It requires a cell to build a cell. And so what we find in the structure of a cell is a template, in effect, which allows individual gene products as they're made to go ahead and assemble in place in order to build the structures that are necessary. Now, you can watch this process in phase microscopy looking at something like these 2 cells shown here, imaged in my lab a long time ago. It's a time-lapse movie which compresses about 20 hours of a cell cycle into about 30 seconds. So time is flying here, and you can watch any one individual cell, and as you watch it moving around, you'll see it divide. The periods between division, which are of course called interphase, are the times of synthesis that I've been talking about. It's then that proteins are made, RNA is made, DNA is replicated, so that we have 2 cells in one bag. The process of cell division, then, is the mechanics of separating all those constituents into 2 discrete objects. And it has to be done very well, because if cells lack essential constituents, for example, a chromosome, some of the DNA, which includes instructions for making RNA and protein, then the daughter cells will be unlikely to survive. So the division process is a very precise one that has to take advantage of structures that are built in order to assure the accurate segregation of the components of the cell, which are present in only a few numbers, like chromosomes. What you're seeing here is cell division going forward with no restraint. The cells are being provided with the factors that stimulate their division. They're being provided with all the food that they need, and so they simply divide as rapidly as they can complete the essential synthetic processes. This is like the lifestyle of a unicellular organism, where food is really the limiting factor in how a cell can go forward, making decisions to divide and produce two of itself. For the cells in our body, on the other hand, this process must be tightly regulated, because if cells divide too frequently, you can get anomalies which are very dangerous medically, like cancer. On the other hand, cells must divide in order to achieve a healthy adult organism. Of course, the cells of an embryo divide in order to produce the juvenile and then the adult form. Cell division is essential in wound healing. Cell division is also essential in tissue renewal. So, for example, the red blood cells that circulate through your vasculature are comparatively short in their lifetime, a couple of weeks, and if they are not replaced, you will have anemia. So cell division is being regulated in a multicellular organism to produce all the cells that are necessary to balance the cell death which is occurring. And this balance of cell division and cell death is a big subject in its own right. That's not what I'm going to be talking about now. What I'm going to be talking about is the ways in which cells go ahead and synthesize during these various periods of interphase the constituents they need in order to be able to achieve an accurate cell division. The synthesis, of course, includes DNA synthesis in the S phase of the cell cycle, but there's a gap before that and a gap afterwards, and both of these are times of continued protein synthesis, RNA synthesis, and cell growth, so that when the cell finally comes to make a decision to enter the division process, it is already fully two cells in one bag. And the division process itself is simply a mechanical division of all those constituents. Now, when a cell is going to try to divide, it faces a series of problems. As I've been emphasizing, there must be enough of all of its components that it can go ahead and provide for the daughter cells. There must be enough ribosomes and mitochondria and individual enzymes and so forth. These are objects that are present in large numbers. And the strategy that a cell has for handling these in division is that if there are enough copies of an individual object, like a ribosome, the chances that one daughter cell will get all of them are very small. And so simply pinching the cell in the middle will be sufficient to assure that each daughter gets plenty. But there are other structures in the cell which are present in small numbers of copies. Chromosomes are an example. The centrosomes are an example also, the organizers for the microtubule component of the cytoskeleton. Here you have one or two copies of each object. They duplicate during interphase, and in order to assure that the daughter cells will get everything it needs to divide, we have to have a special machine that is going to assure equipartition in the time preceding cell division. Now, that process of equipartitioning and cell division is the process of forming the so-called mitotic spindle to segregate the chromosomes. And this is a structure that is familiar to all of us from the spring of our scientific career, and yet it's a wonderfully complex structure that is remarkably accurate in its ability to do its job. It forms in the cytoplasm off of organelles, the centrosomes in animal cells, and less well-defined structures in some other cell types. And it represents then a family of microtubules that will grow into the region of the nucleus where the chromosomes have been condensing. It will then attach to the chromosomes and organize them, and the organization puts them into the structure of the metaphase plate. Chromatids then will segregate and will get then this anaphase process, which involves chromosomes moving to the poles and the elongation of the spindle so that later on the cell can simply pinch in the middle and ultimately give rise to two cells. So the mitotic spindle is the structure which is this special machine. It segregates the chromosomes and it segregates the centrosomes, and that's what we're going to try to understand. The job that the spindle faces is an extremely difficult one because eukaryotic cells in particular have a huge amount of DNA. There are many, many bases, all arranged in long strings, and these strings are in fact long enough that they make polymers that are millimeters to even meters in length, whereas the cell is measured in many micrometers in length. So there is a factor of thousands in the length scale difference between the polymers that we need to segregate and the cell itself, which means that the DNA must be all bundled up in some way within the cell. And these chromosomes, one or more, have to be segregated accurately if we're going to get all the DNA into the daughter cells. And this must occur for the viability of the daughters because the loss of even a single chromosome is generally lethal. You lose so many genes that the cell just cannot survive. This set of micrographs brings us back to the nucleus with which we started, but shows that if you allow the DNA to spill out and you wash it clean of some of its proteins, you can see what a tremendous extent of material it is. This long extent is what we need to duplicate. And our problem is that it's about 5,000 times longer than the diameter of the cell, and how do we achieve this process? The solutions that cells have come up with in order to solve this problem are numerous. One of them is that the DNA is packaged in more than one piece, as a rule, in eukaryotes anyway, and this means that we've divided up all that DNA into smaller segments. The DNA all is replicated before cell division begins. This means that in eukaryotes we have a situation where the whole set of double DNA is ready for us to operate on. Another trick that the cell uses is to keep these sister chromatids, as they're called, the duplicated DNA double helices, fastened together so that the two identical pieces of DNA are linked non-covalently by a protein complex, and it's going to keep them in order while the cell is getting ready to divide. The other point is that the chromosomes will condense tremendously, decreasing their length down to make them an object that is small enough that its full extent is less than the diameter of the nucleus. So there's a tremendous amount of compaction, and then finally we will develop that special machine, the mitotic spindle, which can do the segregation job. Now, when DNA is replicating, what you have is here a piece of DNA which is a circle. This is actually a viral genome, and here is a replication fork. Over here is another replication fork, and the origin of DNA replication would have been here and up here, and this is now duplicated DNA. And these forks will travel apart, making completely replicated DNA. In a eukaryotic cell, the chromosomes are not circular. They're linear, but their linearity doesn't make the problem easy, and I mentioned that the DNA is held together as it is replicated. So here is a replication fork. Here are the sister chromatids, which have been produced by replication, and this is a complex called the cohesion complex, which in some way fastens these sister chromatids, as they're called, together, so that as the DNA wraps on the nucleosome core particles, which is the first stage of condensation, the sister chromatin, which is the name for the material that is DNA plus protein, is held together, and these sisters are associated. Exactly how the cohesion complex does this is still not fully understood, but this diagrammatic representation of it surrounding the two is a plausible way to think about how it could link sister chromatids together. Once DNA replication is complete, condensation will occur, and here are multiple stages of the condensation diagrammed in a textbook form. The condensation is going to give us, in most cells, thousands of fold decrease in length. In some cells, it's not so much. So, as is common in biology, there's a lot of variability, and you have to account for that as you're thinking about trying to understand a process in a simple term. But all eukaryotic cells use this packing on nucleosomes, and the nucleosomes then pack together to form this fiber, which looks like about 30 nanometers in diameter when it's seen in an electron microscope, and then this folds in loops, and then the loops condense, and finally it comes to the chromosome, which name of course means the colored body, and that gets that name because once it's condensed sufficiently, you can see these strands of DNA in the light microscope with sufficient good resolution and stains, and chromosomes were recognized as such even in the 19th century. Now, there's still a lot of work going on to try to understand this process, and proteins have been discovered that were thought at first to be extremely important for it. For example, a protein called condensin, which is involved in making the chromosome become more condensed. But it turns out that that protein is not necessary for the condensation process. It probably depends instead on a combination of post-translational modifications of the proteins that associate with the DNA in order to compact the chromatin by changing their charge and probably changing the proteins with which they associate. So this condensation will bring us to the time when we can enter division itself. And this is a still frame from a movie taken by my friend Jeremy Pickett Heaps of a newt cell in the process of cell division. The nuclear envelope is still intact, and you see the condensed chromosomes here within the nucleus. And as I run the movie, what you'll see is that the nuclear envelope breaks down and something now affects the chromosome behavior. Chromosomes appear to be moving and becoming organized. And this is, of course, the process of moving towards metaphase. It's the stage called prometaphase, and the process of moving chromosomes to the metaphase plate is called congression. So they're gradually getting lined up, but it's clear that there are also renegades that don't get in line in time, and some of them will even depart and then go back, but eventually anaphase will start. Anaphase is this process of ordered segregation followed by cytokinesis, where the cell pinches in the middle in animal cells. In plant cells, you build a wall between the two daughter cells instead. But what this gives rise to then is two independent nuclei, each with a complete fabric of chromosomes, and they're now divided into two distinct cells. Now, in the movie that I just showed you, you saw chromosomes and their behavior, but we didn't see anything about the mitotic spindle, which I said in the diagram a while ago is responsible for this event. The mitotic spindle can be visualized in living cells in a variety of ways. Historically, the most important was the use of some optics that involved polarized light. And a number of people, principally Shinya Inoue, have been responsible for taking advantage of polarized light in order to visualize the machinery for chromosome movement as it acts. This is a movie that I'm going to show you which is taken with a brand new kind of polarization microscope, which gives a brightness reflecting the way in which polarized light is interacting with the cell in such a fashion that it doesn't depend on the orientation of the object relative to the plane of polarization. And this turns out to be important for getting a clear image. And now brightness is the polarization optical image of the spindle. This is the same cell type you were just seeing, and there's the same anaphase. And you can see that the spindle shortens as the chromosomes draw up to the poles, but the whole structure also elongates in order to give you the greater separation of the chromosomes that we saw at the end of that phase microscopy movie. Here's a diagrammatic representation, although this is actually an electron micrograph taken of a mammalian cell in culture which was fixed and then stained with tubulin using colloidal gold to bring out the microtubules that are here. The chromosomes are evident just from their own binding of stain. And here I've diagrammed with my arrows where the poles of the spindle are, where the chromosomes make contact with fibers that come from the poles, and the special region on the chromosome to which these fibers attach is called the kinetochore. This comes from the Greek root for meaning movement. Out here we have fibers which extend radiating out from the poles. These are often called the astral rays, and they're common in many animal cells, but they're by no means universally found. They appear to be part of the process that will center the spindle within the cell and may be part of the elongation of the spindle process, but they're not essential for organizing and segregating chromosomes. This micrograph gives us an overview of what the spindle looks like, but we can look more deeply into the spindle by means of higher resolution electron microscopy, here cutting thin sections to see the kinetochore as a layer of proteinaceous material which is stuck onto the underlying chromatin, which is here. Microtubules are coming out from this kinetochore, and this attachment is a key part of forming a functional spindle. The spindle in many cells grows from a structure called the centrosome. The centrosome contains two centrioles, so-called, in many animal cells, and some pericentriolar material. This includes a special isoform of tubulin called gamma tubulin, which is held in place by a series of other proteins, long fibers like pericentrin, which are holding the gamma tubulin in place, making a series of microtubule nucleating sites from which tubulin can polymerize to make the microtubules of the spindle. This is what a cell looks like as it goes into mitosis. Again, an electron micrograph with a chromosome shown very dark, the spindle poles at the ends of the spindle, and you can see that the distance between the chromosomes and the poles has begun to shorten. This shortening is called anaphase A, and it's an essential part of the segregation of chromosome process. The spindle elongation is called anaphase B, and it leaves behind this interzone in the middle, which appears in this micrograph to be essentially empty. But this is simply because those microtubules are not being stained in such a way that they show up here. There are actually microtubules there. And I can show you this, again with polarized light, looking at a meiosis spindle, meiosis 2, which is mechanically very much like a normal mitosis. And this is in the spindles of a crane fly, which is a particularly photogenic object. So again, with polarized light, we'll see here two cells that are the products of the meiosis 1, and they are now each going to form their own mitotic spindle, which will then organize the chromosomes, and you now see the chromosomes as ghosts on the spindle equator. Anaphase will start, and the chromosomes are now going through anaphase A and anaphase B, with the birefringence in the middle of the spindle giving us an indication of the amount of structure that's there as these 4 spermatids are produced from this second meiotic division.